This is Jim Schnabel doing talk number 29 in the series from Principles of Biochemistry by Albert Lehninger, 7th edition. Today we'll be discussing the biochemistry of cholesterol, steroids, and isoprenoids. Cholesterol is the critical component of membranes, is a precursor of steroid hormones and bile acids, and is correlated with atherosclerosis. Cholesterol has an acetate precursor. There are 4 steps of synthesis with isoprene subunits. That is the skeleton that we see in the biologically active hormones. 3 acetates are condensed to form 6 carbon unit mevalonate. Mevalonate is converted to isoprene. 6 isoprenes are polymerized to squalene. So we have 30 carbons. And then the cyclization to a 4-ring steroid, and then subsequently modified to cholesterol. So when we look at this, we have 3 acetates, and they combine to form the structure mevalonate. There's an activated isoprene, which is here, and you get this structure which condenses to form squalene, which is in this the linear form, and then the rings form and you get cholesterol. This is the structure of cholesterol. This is obviously a broad schematic. If you look at it more closely, you have 2 acyl-CoAs. There is an acyl-CoA acetyltransferase with liberation of CoA. Here you get 2 of these cleaved together as acetyl-CoA, acetoacetyl-CoA. Then another CoA molecule is added to this. It combines at this point right here, this carbon, so you get this structure here, which is beta-hydroxy, beta-methylglutarryl CoA, or HMG CoA. Then you get a reduction with liberation of CoA, and you get from that mevalonate right here, from this to this, with reduction of this carbon, number 5 carbon. Mevalonate then undergoes phosphorylation. You get a 5-phospho mevalonate with this structure here. Another molecule of ATP is attached. You get a pyrophospho mevalonate formed there. Another ATP gives a phospho right here to number 3. So there's number 5 pyrophosphate attached. Number 3 has phosphate attached, and this causes elimination of carbon dioxide using this as a fulcrum or an electron sink. So we lose the phosphate, we lose this CO2, and we get a delta 2, or rather a delta 3 isopentenyl pyrophosphate right here. And then there's an isomerization that occurs, and then you get dimethyl allyl pyrophosphate right here. So here's the isomerization part right there. Right in this spot. This reaction continues, and you have dimethyl allyl pyrophosphate, delta 3 isopentenyl pyrophosphate, and these combine with loss of the pyrophosphate moiety, and you get geranyl pyrophosphate. This undergoes a further loss of pyrophosphate, and you get farnesyl pyrophosphate from this. And then the farnesyl pyrophosphate is reduced, loss of pyrophosphate, and you get squalene. Squalene is the next molecule, the linear form. And then squalene is further reduced. You have loss of oxygen, loss of water, formation of an NADP, and you get a squalene 2,3 epoxide. And this can either in a multi-step in plants go to stigmasterol. You can have an intermediate that goes through to linosterol and then cholesterol, or in fungi you can go to ergosterol right here. So there are three possible fates of squalene 2,3 epoxide. These aren't necessarily complicated structurally speaking, but the chemical reaction mechanisms are fairly straightforward. For cholesterol metabolism, they're mainly synthesized in the liver, exported as bile acids, cholesterol, or cholesterol esters. These are converted to steroid hormones in gonads and adrenal cortex, and vitamin D in the liver and kidney. Cholesterol is transported in the blood as a lipoprotein. Apolipoprotein has a protein moiety, and these are signals from metabolic action. Apoproteins appear in differing amounts in lipoproteins and have different functions. This is the composition. As stated previously, if you take plasma and centrifuge it, the creamy layer on top is generally completely comprised of triacylglycerols, and this is the chylomicron layer. As you further centrifuge and get further fractions, you get VLDL, and as you go through the sequence from chylomicrons to VLDL to low-density lipoprotein to high-density lipoprotein, the density not only increases, but the amount of protein increases, the phospholipids increase, and the triacylglycerols drop in their relative composition. The entities which are cardiogenic are LDL and VLDL. These are the two that comprise so-called bad cholesterol, and chylomicrons are generally seen after a meal. HDL is the so-called good cholesterol, and this is increased under certain physiologic conditions such as exercise. This is a schematic of what happens in the body. You've got macrophages and foam cells here. The first thing is that the fat, which is present in the intestine, is transported out through specific sites. You get chylomicrons that enter the lymphatic system. You have markers, apolipoprotein B, apolipoprotein E. They can enter the capillaries. You get free fatty acids forming, chylomicron remnants, and these free fatty acids can be utilized in the mammary gland. They can also go to adipose tissue, muscle, adrenal gland, and also be transported to the liver through the lymphatic system and the circulation, but mainly the circulation. So the venous circulation from the intestine brings these to the liver, where you can get an enterohepatic pathway where bile salts are stored in the gallbladder. They're released from the gallbladder into the small intestine at the junction between the stomach and the intestine to emulsify fats. You also can have cholesterol with cholesterol esters be transformed to VLDL, which can enter the circulation. You can also get LDL produced, and that can be peripherally metabolized and formed and go to LDL receptors in the liver. You also have production of HDL, and you can get those produced, and they can eventuate in macrophages and foam cells, which is the end-stage repository for LDL and HDL. So physiologically speaking, excesses of LDL and VLDL are correlated to atherosclerosis, which HDL has a protective effect. So again, the liver produces VLDL and can be transformed to LDL. You get LDL going back to the liver. Reverse cholesterol transport can take HDL and move it back to the liver. And the idea is that the deposit of the cholesterol and the cholesterol plaque is a complex where you get a fibrin cap. Monocytes are attracted to the oxidized lipoproteins that aggregate and stick to the extracellular matrix. The monocyte differentiates into a macrophage, and the macrophage ingests lipoproteins and causes a foam cell, which you can see under the microscope. These will accumulate. You get breakage or cleavage of the endothelial lining with attraction, so there is an inflammatory response that helps exacerbate this, and you can get a plaque, a soft plaque. You can get a hard plaque where this ultimately calcifies, and any sort of injury that can affect this lining can break off part of this plaque. You can get thrombosis, you can get myocardial infarction, strokes, and this is part and parcel of the end result of atherosclerosis, with vascular damage and damage to cells, with infarction being a localized area of death within an organ. The exogenous pathway is intestine transport to the liver. The endogenous pathway transports has liver transformation from VLDL to LDL to tissues with the reverse return. And this regulates the fate of HDL. LDLs transport into the cell by specific membrane-bound receptors. Cholesterol synthesis is tightly regulated. The rate-limiting step is conversion of HMG-CoA to mevalonate. This is activated by high ATP concentrations and inhibited by low ATP concentrations. And the high cholesterol causes transcriptional regulation of HMG-CoA reductase and decreases the number of gene copies. And you can also get transcriptional regulation conducted by sterol regulatory element binding proteins. Steroid hormone synthesis is also involves cholesterol. This is a precursor molecule which requires side chain cleavage and oxidation steps in the mitochondria, so you can get cholesterol going to pregnenolone, then progesterone. From progesterone, you can get testosterone. You can transform this to estradiol. So male and female sex hormones are here. They influence secondary sexual characteristics. They additionally regulate the female reproductive cycle. You can also, from progesterone, get corticosterone, which is a mineralocorticoid, which is involved in salt regulation, which is aldosterone. This is the end result. It regulates reabsorption of sodium, also chloride and bicarb in the kidney. Cortisol is also formed from progesterone. This is a glucocorticoid, which affects protein and carbohydrate metabolism, suppresses the immune response and inflammation and allergic responses. And the intermediates that occur and also produce vitamins, plant pigments, and fragrances. And that will do it for this talk. Thank you very much.